there will be some questions and answers from your quiz, and I will explain a little bit more some of these topics. Okay, so let's start. So, question that is very easy, you can read it with me. What is the primary difference between a 2D and a 3D tissue culture? So it's pretty easy. So you can read the options, the type of media used to grow the cells. So you can stop this for a second and pick the answer. Stop the video and I will give you the answers and the explanation. Okay, so I think you already know the answer, but so you know that the first one is that a 2D and a 3D, just 2D will be a tissue culture that is in a petri dish, and 3D culture, so the cells are flattened here in this culture, yeah, in a 2D fashion. A 3D, it will be a structure that has 3 dimensions. So the media is not the problem, yeah, so this is not the problem. The number of cells is not the problem, yeah, it doesn't make something 2D or 2D or 3D. The dimensionality of the culture system is the key difference, and the type of cells is not the problem. Okay, so we are going to see that these 3D cultures, and as you watched in the previous video lecture, um, they are fundamental to study the extracellular matrix. And actually, a person who started working on these 3D cultures, they she said that if you remember the video lecture, she said that she said that she didn't patent the 3D cultures, those because they made a lot of money. The people that make a company based on 3D culture. So let me show you the answer. So it's the dimensionality of the cultures, and I will show you a video of a company that talk about the benefits of having a 3D culture and the difference. So in the next slide, I will show you the video.
Three-dimensional 3D cell culture is defined as the cultivation of living cells within microassembled devices and supports with three-dimensional structures that mimic tissue and organ-specific microarchitecture. Cells naturally grow, mature, and differentiate in a three-dimensional environment, just as they would in vivo. The use of 3D culture is an accurate method of replicating this process in vitro. When compared to other cell culture methods, 3D cell culture is becoming more popular due to its novel and convenient features. In contrast to 2D cell culture, 3D cell culture promotes cell differentiation and tissue organization through the use of microassembled structures and a complex set of environmental parameters. In fact, cells grown in a 3D environment are more susceptible to morphological and physiological changes than those grown in a 2D environment. In addition, 3D cell culture also allows for the simultaneous growth of two different cellular populations, with co-cultures accurately reproducing cellular functions observed within a tissue, as opposed to co-cultures based on 2D cell culture. Finally, thanks to microengineering, 3D cell culture enables greater control and monitoring of the microenvironment parameters of growing cells, temperature, chemical gradients, oxygen rate, pH, and so on, while remaining as close to reality as possible. While 3D cell culture may be a cost-effective technique that could eliminate the need for animal drug testing in clinical trials, developing automation and reproducible applications remains a time-consuming and meticulous process.
Okay, so here is the main idea of the video. Explain that I want that you remember when you have cell culture. Great technique. Remember, it's an in vitro approach, and you can study the context of the cells, the contact with the surface. However, if you see here, there is not extracellular matrix. Yeah, so there is not SEM, and if the shape of the tissue play a key role in the function, here you're missing that. Okay, so here what we were talking is that the cell culture is a widely in vitro tool for improving understanding in several fields like cell biology, tissue morphology, and different mechanisms of drug action and production. However, um, although in the past cancer biology was used a lot two-dimensional cell culture, there are some limitations as you watch in the video, and as I explained uh, the previous slide, you have interactions with the extracellular matrix, and as you are changing the morphology, this can create some bias. So to solve these problems, the main difference, remember, is the dimensionality. You can have the cells in an extracellular matrix, and here the tissue can grow more similar to what happened in the in vivo case. And so these 3D cultures, for answering some questions in biology, in particular in cancer biology, they have been very useful. And every time we try to make something that looks more similar to the in vivo, so the in vitro is great because you have more control and you have less variables. So, but so the 3D it is like an intermediate. You have a little bit more variables and it's similar, more similar to the in vivo. Right now, maybe you have heard about organoids, that they are not exactly organs, they are not exactly in vivo, but they are in between the 3D and an in vivo experiment. And again, has advantage and disadvantage. So conclusion, this is a 2D culture, this is a 3D one. This one doesn't have an extracellular matrix, this one have extracellular matrix, and these both are very good to use in a specific to answer specific questions. Okay, so possibly you already are tired of these questions, and I'm tired too, but this is a very important pathway. So that again, if RAS is there, you need to memorize things. Yeah, you need to be aware that they exist because they will help you even to read or to write the paper that you have the to read the paper for your presentation or to write the paper, the final paper for the class. So remember, let's read it together. Which of the following is the protein kinase upstream of the MAP in the MAP kinase pathway? So here you have these four options. You can stop the video right now, and even if you want, here you will find the answer. And so stop the video, think 2 seconds, and then I will give you the answer. Okay, so the answer definitely is not the JNK. So the JNK pathway is involved in the cell death. This is involved in mitosis, remember, proliferation, growth, when you have RAS. So although these pathways, they interact, there is a cross interaction between this pathway, definitely this is not, this is the opposite. This is related to cell death. P38 is related to something different, so no. And these two are definitely part of this MAP kinase pathway, but they are asking you which one is the one that is upstream. So upstream, it will be the one that is higher. And here you can see that MEK is higher than ERK. Yeah, both are correct, but the one that is both belong to this pathway, but the one that is upstream is here. If they were asking you and the one that is downstream, the one that is downstream here in this case will be ERK. Okay, so pretty easy. And if you remember the video of the beginning of the class or the one that is in Moodle, you can rewatch it and see in more detail this question about the MAP kinase pathway. Okay, so remember, this is the correct one, and here you can see how the phosphorylation of this MEK that is upstream and downstream will be ERK. Okay, and then as usually there is a change in the transcription when it goes to the nucleus. Okay, so let's go with to the next question. Okay, so this is pretty easy. What is the main benefit of target and control approach to cancer treatment? Okay, so if you watch the video, this is a question that is very easy, and they talk about different types of treatment of cancer, and that we're moving from one to another one. So please stop the video, give it a read, and later I will go and try to answer the question, try to help you. Okay, so I imagine that you already know the answer, and let's go for A. It is a higher success rate than seeking and the destroy approach. Yeah, is not. It could be a good answer. Maybe it's not the best one. It's not always. It doesn't have all. It's not always they have a higher success. Hopefully, it will be. It always be true, but it's not. It's not completely true. Okay, let's go for B. It is more affordable and accessible that seeking and destroying approach. Okay, so right now is not. This is not true. Like these require a lot of research. So right now is not cheaper and more accessible either. So not affordable, more affordable in the present, no, and more accessible. C, it allows for target, targeted and personalized treatment. So remember, this could be uh, this could be the best answer because remember when you target and control approach instead of attacking all the tumor, all the cells of the body that have many side effects and the patients are not have so many like a really bad time because you're attacking all the cells of the body, not only the ones that are involved in the cancer. This is really tough for patients. So based on the patient's specific cancer and type of genetic profile. So this is it seems to be a better answer. This seems to be the best answer. It's less invasive and has fewer side effects than the seek and destroyed. Yeah. So this could be true. So B is definitely wrong. A could be true and D could be true. However, the best answer is definitely C because it allows for target and personalized treatment based on the specific cancer type and genetic profile. So this, if you remember the video, we are saying that we were going to a video from an approach that it was like chemotherapy. It involved many times attacking all the cells from the body to try to kill the cancer cells, but you're also killing the normal cells. This is why they lose the hair. There are so many side effects. The body is not feeling like the patient is not feeling well. So the idea is to move from this one that you're attacking all the cells to just go and attack the cells, the cancer, based on the on a more uh, in an oriented response, attacking those cells, attacking that particular tumor. So it become more personalized because some treatments, if you maybe they don't work for some people, while other treatments work for other people. If you remember the video lecture, she was talking about how there is a variety of tumors, that somebody of genetic profiles of the tumor, so that somebody has lung cancer doesn't mean that all the cancers, all the treatments for lung cancer will be the same. And so this is a very good, a much better approach that hopefully we can work on that. To finish this section, so remember, for extra credit, if you want an extra credit, if you want to have more control and you're so like, oh, Nicholas is talking about so many things, you can read this review paper. Yeah. The textbook right now we are going to stop using because the next sections are more involved to research, so they are not still in the textbook. The textbook is great, they just don't have the new information is not yet in the textbooks. Here there's two papers. You can read each one. One is more recent, the other one is a little bit older, um, but is very clear. So you can pick each one of those. And here you can see again junctional remodeling of a cell. And here we will focus on these two type of cells, or two type of proteins. So give it a look if you want to have a more control over the topics that we are going to cover.